all for coming. Thanks again for coming. It is my pleasure to be able to introduce, I guess he's above me in the little pictures right now, Vinny Lynch. He got his bachelor's from the University of Albany, did PhD and postdoc at Yale, working with Günter Wagner. We just established it was about 10 years of Günter, which is a lot of Günter. It's good Günter, but it's a lot of Günter. And is currently a PI in the Department of Biological Sciences at the University of Buffalo. Vinny has over 50 publications, just truly beautiful work in a lot of different areas. And from his site, the description is that they use comparative genomics and experimental methods to study the evolution of pregnancy, the evolution of animals with extremely long lifespans and large body sizes, and the role of evolutionary history in animals' susceptibility to preterm birth and diseases like cancer. So with that, I will welcome and turn over the floor to Vinny.
Well, thanks for the introduction, Karen. And thank you all for being here. I'll just note that I don't mind being interrupted during talks. Particularly by grad students and postdocs. Chances are, if you don't understand something, I've either said it wrong or have just gone too fast. I tend to talk fast, especially the more I get excited about something. And if you don't understand something, chances are your friends don't understand something either. So like, do them a solid. And if you have a question, just stop and ask me. So the lab sort of.
So I should say, sorry, for one second.
No,
so in order to ask a question, either raise your hand, not physically, but with the clicking button and the participants, because we can't see your faces, or you can put something in the Q&A if you want. Okay, good.
All right, good. All right, that didn't work. Okay, so I consider myself a Evo person, which just means I'm interested in sort of how development influences evolution and vice versa. And generally within the sort of broad field of evolutionary biology, basically two research programs, right? You either study adaptations or you study speciation, right? So Darwin wrote the book. It's called On the Origin of Species, but he doesn't really write much about how species originate, but he does talk a lot about how adaptations happen and sort of tries to come up with an explanation for evolutionary change, focusing a lot on adaptations. And depending on how you count, over the last 30 years or so, there's a third research program which has sort of emerged or reemerged, depending on who you talk to. And the goal of this research program is explaining how morphological characters evolve. So those characters need not have any effect on fitness, so they don't have to necessarily be adaptations, and they don't necessarily have to have an effect on speciation, so they can be independent of that, right? So this research program is principally interested in understanding morphological evolution for morphological evolution's sake, regardless of whether those things influence speciation or fitness, right? So we blame Darwin for evolution by natural selection and speciation, and we generally blame Devo-Evo or Evo-Devo, depending on who you talk to and which camp you belong in, on sort of early comparative anatomists, in this case, Richard Owen. So while it's pretty clear that sort of the molecular mechanisms that underlie morphological evolution are sort of debated, at least that debate happened a while ago, it still sort of rises up every now and then. The precise mechanisms are debated, but we all basically know that the way the genome evolves ultimately underlies the way morphology evolves. And the debate was usually between people who thought that all of the important kinds of evolution occurred in cis-regulatory elements, enhancers and promoters and silencers and the like. And then the rest of us who thought that, cis-regulatory evolution played a role, but, transcription factors evolved too. And if you were smart, then you believed that both of these things contributed. Of course, you were just sort of stuck in the middle, like this poor little spider, and you were likely to get trampled on. But regardless of whether you're sort of in the cis camp, the trans camp, or somewhere in between, I think we all agree that if we want to understand how morphology evolves and sort of do the Devo-Evo thing, then we really need to understand how genomes evolve, because genomes give rise to morphologies through the process of development. So if we want to understand any of that, we at least need to understand how genome evolution works. And for us interested in Devo-Evo, This particularly means understanding how genomes evolved during the origin of sort of big morphological transitions. We generally think of these transitions as being like the origin of novel characters. If you're a mammal, you have lots of novel characters, right? You have hair, mammary glands, pregnancy. The origin of innovative functions. So, you know, the function of a feather now is to fly, but ancestrally that was probably to stay warm. So you have all these sort of morphological traits, right? So we really want to understand how genomes evolve during the origin of morphological transitions to understand how development influences those transitions. So that's all well and good, but for all things you need a model, right? So one of the nice things about being an evolutionary biologist, as probably you all agree, is that everything evolves. So it allows you to have sort of an intellectual ADD and study everything, which is fine. But you still need a model to understand the mechanisms of things. And one of the models that we've been using is the evolution of mammalian body size and lifespan, right? There are lots of reasons to focus on body size and lifespan. And evolutionary biologists have been doing it for a long time, right? So these are pretty critical life history traits. And what makes them nice from an evolutionary point of view is that there's a lot of variation in both body size and lifespan across a whole bunch of different vertebrates. Some of you may have noticed that that's not a vertebrate, but let's just go with animals. So there's a lot of variation in body size and lifespan in animals. So just among the vertebrates, body size ranges from, you know, gigantic blue whales to tiny, tiny salamanders. And I swear there's like a pixel dot in that corner, right? So one of the smallest vertebrates that we know of is the eastern redback salamander, and it weighs about half a gram. And then the biggest living vertebrate is the blue whale, which is clocking it at 136,000 kilos. So there's a huge variation there in body size. There's also a lot of variation in lifespan. So the oldest vertebrates that we know are these Greenland sharks. They live in the Arctic waters of Greenland, obviously. And we know that they can live more than 400 years, but we're not really sure sort of how much more than 400 years. But 400 years is a long time, even if that's like the max age that they can evolve to. So that's the upper limit. The lower limit are these fish which tend to live in sort of annual pools. So things like African turquoise killifish and these pygmy gobies, which live a few months to, maybe half a year or so. So both body size and lifespan vary pretty dramatically within vertebrates. And of course, these things are correlated, right? So if you're big, you live a long time. And if you're small, then you don't live a long time. So there's a very strong positive correlation between body mass and lifespan. And while we know that there are a lot of constraints on the evolution of large bodies and living a long time, at least one of them we think is an increased risk of developing cancer. And the logic goes something like this. So this is a thought experiment at the moment. It's not really based on real data yet. So imagine that you have some small animal and some large animal. And now imagine that for both the large animal and the small animal, the probability that any cell transforms from a normal healthy cell into cancer is the same. So it's the same for small things and it's the same for big things. Then we think that on average, big things, which have more cells than little things, should just have more cancer, right? So the bigger you are, the more cells you have, the more potential you have for any of those cells to turn into cancer. And the same logic is true for lifespan. So if the probability of developing cancer is just the waiting time to acquiring cancer-causing mutations, then things which live a long time just have more time to acquire those cancer-causing mutations than things that don't live a long time. So all else being equal, long-lived things should have more cancer than small-lived things. So that leads to an expectation, right? So we would expect then that big things like elephants and whales have more cancer than intermediate-sized things and have more cancer than the smallest thing. So there should be a really strong positive correlation between body size and lifespan and cancer risk. And if you look around the world, that actually tends to be the case. So for example, in humans, tall humans have more cancer than average height humans. In particular, they have more, so it varies by the kind of cancer, but for example, they have more colon cancer. So the risk of having colon cancer throughout your lifetime is greater if you're taller than the average. And the reverse is also true. So if you're shorter than average, your risk of developing across your lifetime of developing cancer is reduced, right? So there's a within humans, there's a pretty strong positive correlation between height, not body mass, but in this case, height, and the risk of developing cancer, right? So we see that sort of expectation come true in humans. The problem is that humans don't actually vary all that much in their sort of non-pathological body sizes, right? So the tallest humans aren't actually that much taller than sort of the shortest humans. If you sort of compare that size distribution to the distribution of mammalian body sizes, right? So the biggest mammals are way bigger than the smallest mammals, but the biggest humans aren't that much bigger than the shortest humans. So we don't actually have a lot of data from within species comparisons, where within a species, there's a lot of variation in body size. But we do have it for some things. And one of those species that we have is dogs, right? So if you look at the incidence of cancer for different dog breeds that have different body masses, in this case, it's 70 different breeds and just under 16,000 individuals, there's a pretty strong positive correlation between the body size of that breed and its incidence of developing cancer. So the biggest things, and by here, big dogs, think of like St. Bernards and Irish Wolfhounds and the like, nearly 50, 60% of them will die with cancer. Whether the cancer killed them or not sort of depends, but they'll all die at least 50 or 60% with a tumor of some kind, usually bone cancers, osteosarcomas. Meanwhile, the smallest dogs, actually, almost none of them develop cancer. So here, think of things like Chihuahuas and teacup Yorkies, and actually the smallest dogs, which sort of have zero. So the smallest dogs almost never have cancer. Well, the biggest dogs, cancer is very, very common. So within species, we have the expectation that body size and cancer rate should be correlated. And that's what we observe, right? So within humans, there's a strong positive correlation between body size and cancer rate. And within dogs, there's a pretty strong positive correlation between body size and cancer rate. So that leads to an expectation, right? If you observe a phenomena within a species, then you expect to observe that same phenomena between species. And the problem then arises that if you look at cancer rate by body size in a whole bunch of different eutherian mammals in this case, which vary a lot in their body size, there's almost no correlation, right? So elephants are this dot here, right? So elephants are way bigger than sort of grass mouse, a very small mouse, but they don't really have that much cancer than them. So in the pink dots are data for 37 different species of mammals in the San Diego Zoo, so nearby you guys, and it's just under 1,500 individuals. And if you look at the body mass by cancer rate, then there's almost no positive correlation between body size and cancer. There are a couple standouts or outliers like this dot. So this dot are, what is that? That's Tassie devil. And they have that transmissible cancer thing. So let's exclude them because transmissible cancers are sort of a weird kind of cancer. And this other outlier dot are cheetahs. Cheetahs are incredibly inbred, which is probably increasing their predisposition to lots of things in addition to cancer. So if you sort of exclude those outliers, and then if you sort of see absolutely no correlation between, maybe a slight negative correlation if you squint, between body size and cancer risk. And one of the first people to recognize this was Sir Richard Pettos. So he was a cancer epidemiologist studying the effects of smoking on lung cancer. And what he recognized, at least in the samples that he was studying, is that the cancer incidence of the mice in his lab colony were about the same as humans. And he recognized that, well, humans have more cells than mice, so they should just have more cancer than mice. So we now call this expectation of finding a positive correlation between body size and cancer, but not finding the correlation between body size and cancer between species, Pettos paradox after Petto. And paradoxes are really interesting things from the point of view of an evolutionary biologist, right? Because they demand explanations. So there's been a lot of work trying to understand how Pettos paradox has been resolved during evolution. So why is it that we expect animals with large body sizes and long lifespans to have a greater incidence of cancer, right? To have more cancer than the smaller things, but not, right? So clearly, at one level, the explanation for how these big long-lived things reduce their risk of developing cancer over their lifetime, it's sort of trivial, so trivial as to, well, not be all that interesting, right? So clearly some lineages, the big ones and the ones that live a long time, they basically broke whatever developmental constraint there was on the evolution of their long body sizes. And they probably did that by just evolving, well, by evolving better cancer protection mechanisms, right? So if you look at all these animals and sort of look to see which ones probably evolved better cancer protection mechanisms, a few stand out. I like elephants, so obviously we'll talk about elephants, right? So clearly elephants, which are giant, for whatever reason, evolved mechanisms that broke the developmental constraint on gigantism, right? So you have really big African elephants and Asian elephants. They're big, but the sort of extinct mammoths are even bigger than the living African elephants. And African elephants and Asian elephants and mammoths, so their closest living relative is a hyrax, which is this thing down here. And hyraxes are pretty small. They're about the size of a groundhog, right? So ancestrally elephants are groundhog size, and they got to be giant by sort of evolving better cancer suppression mechanisms. And obviously cetaceans, right? So whales and elephants are really, elephants are big bodied, but whales are even bigger bodied. So clearly whales and dolphins must have broken the developmental constraints on sort of evolving their large bodies, right? So blue whales are giant, porpoises are big, dolphins are big, but they're all closely related to hippopotami. Hippopotamus, right? So there's a big difference in body size between a hippopotamus and a blue whale. Blue whale probably evolved better cancer suppression mechanisms. We don't really think about snakes all that much in my lab because they're not a mammal and I'm pretty mammal-centric, but there are some pretty giant snakes, right? So snakes vary in body size from the reticulated python, which is on average about 33 feet long, but they're like that, that big around. And the smallest snakes are a few inches long and maybe as thick as a piece of spaghetti. So there's a lot of variation in body size of snakes. And then clearly birds, right? So there's a lot of variation in body size in birds. The biggest birds now are like the paleognaths, so ostriches and rheas and the like. But the biggest birds were the elephant birds. So this one here, which had a height at the shoulder of about the same size as an Asian elephant, right? So there are some pretty big birds in the past. And that picture I showed in the first or second slide of Richard Owen standing next to this leg is the elephant bird leg, right? So the elephant bird, top of the elephant bird leg is about as high as Owen was, and he was like 5'6" or so. So there are lots of variation. There are lots of species which vary in body size, right? So then the obvious question is, so of all these species that vary in body size, which lineages are a good model system to sort of explore the correlations between genome evolution and morphological evolution as it relates to, of course, let's say, remind me later. Close. Bring my shared window. There we go. Right, so there's a lot of variation in body size. We need to pick a lineage if we want to explore the genetic basis of that, right? So what lineages are a good model system? So for lots of reasons, we focused on mammals because mammals vary a lot in their body size. We also understand a relatively lot about the genetic basis, the genetic basis of cancer in mammals because it's been most well studied in mammals. And if you want to get funded, the NIH probably will be more likely to fund, is more likely to fund the studies that explore the evolution of cancer in mammals as opposed to the evolution of cancer in jellyfish or snakes, right? So we focus on mammals for lots of reasons. In particular, we're interested in understanding the genetic basis of morphological evolution. There's also a whole lot of mammalian genomes that are available and more are becoming available sort of every day. The last time I checked, which was a couple of weeks ago, there were 944 mammalian genomes available. So one of the first things we did was try to understand using the data, so doing the inductive thing of saying, okay, what lineages stand out? So you can look at all those mammals that I was showing in the graph before and say, well, yeah, whales stand out, elephants stand out. But that's sort of cheating, right? So we're sort of looking around the world and seeing the big things and saying, well, the big things are interesting. So we wanted to take a sort of more agnostic approach and say, okay, can we use the tools of evolution to say, to identify the interesting mammalian lineages to explore these questions? And so the first thing we did was generate a time-calibrated supertree of mammals, right? So there are only about 2,200 species of mammals. So we took two mammalian phylogenies from Puttick and Thomas and Beninda and Emmons, and we combined them into a single phylogeny, and that method is called the supertree approach. And in total, that ended up giving us just under 18,000 mammals, right? So in this phylogeny, this is a time-calibrated tree. Each one of the lineages at the tips, each one of those branches, is one of the mammals, one of the 1759 mammals that we have. And because they're mammals, we understand a lot about them, including their body sizes. So we went into the literature and we grabbed data on the maximum body size for that species. So that's what these dots are, right? So the bigger the dot, the bigger the body. The more intensely colored purple it is, the sort of bigger the body that is. So you can see that on this side of the tree, there are a bunch of big-bodied things. On this side of the tree, there's a bunch of big-bodied things. So once you have a phylogeny that relates how species are related to each other, and you have data for some character for that thing, in this case, body mass, then you can use methods like ancestral reconstruction to reconstruct what ancestral lineages, that character in ancestral lineages, in this case, body mass and ancestral lineages. So when we do that, it's sort of fairly obvious that the big-bodied things inherited their big bodies from big-bodied ancestors. So all of these extant species which have big bodies have big-bodied ancestors. Same thing for this side of the tree. All of these big-bodied things have big-bodied ancestors. The common ancestor of mammals was relatively small, so sort of tree shrew-like or squirrel-like. So clearly mammals started off small and got so once you have time-calibrated tree, right? So you have branch lengths in proportion to time, like this. You have extant character states and you have ancestral character states. Then you can start to ask the more interesting questions, right? So you can transform this tree from being a time-calibrated tree to being a rate of change tree. So now instead of the branch lengths being drawn proportional to time, they're drawn proportional to the rate of change. So clearly this branch is really, really long. So the rate of body mass change was really, really fast in this lineage and relatively short, relatively slow in all these lineages. So then some lineages obviously stand out, right? So this group that has the really, really long branch and therefore has a really rapid rate of body mass evolution is called the Ferungulata. So this is basically carnivores and things with hooves. Cetaceans obviously stand out, right? So the biggest mammals are cetaceans, so they have really long branches, indicating they evolved to those large body sizes relatively rapidly. On the other side of the tree are the animals I care about, the Proboscideans, right? So elephants and their relatives got their large bodies relatively fast. So elephants are usually huge unless they get stuck on islands, and then their body sizes decrease rapidly. So you can see this long branch leading to the dwarf elephants, and dwarf elephants are about the same body size as big dogs, more or less. Elephants are most closely related to hyraxes. You can see there's a couple of big-bodied hyraxes, but most of the extant hyraxes are small. And in addition to hyraxes, elephants are most closely related to sort of manatees and dugongs, the sea cows. So that's these species. They're big, but they're not elephant big, except for the extinct stellar sea cow, which is a really large, a really large dugong, basically. It's about the size of an elephant, and it lived in the sort of far north around the Aleutian Islands in Alaska, and basically we probably caused it to go extinct. Then there are bats. So bats are interesting. They have a really long lineage here. Although most bats are relatively small, there are multiple instances of bats evolving large bodies relatively fast. Bats are also interesting because even though their body sizes tend to be small, they live for a really, really long time, which is an unusual combination. And then on the other side of the tree, you have big-bodied rodents like beavers and capybaras. So for the rest of this talk, I'm just going to talk about what's going on with elephants and their relatives, even though we have projects on bats and cetaceans. So what we'd really like to know are, or the question that we really like to ask is, do elephants respond differently to the kinds of genotoxic stress that we know will eventually give rise to cancer? Obviously, we can't ask that question, right? We're not going to be allowed to, keep a colony of elephants on the quad and then sort of sort of expose them to different cancer-causing agents and then do survivability studies where every few years we go back and check whether they have cancer or not. Elephants also live a long time, right? So they live 60, 70 years. So I'm 40. Chances are if I were even able to do such a study, I wouldn't live to see the end of it, right? So we can't practically ask those kinds of questions. We're fortunate in that cancer is a cellular process, however, so it's the process of a normal cell turning into a cancer cell. So while we can't ask the question, do elephants respond differently to the kinds of stress that give rise to cancer, we can ask how their cells respond to the kinds of stress that cause cancer. So what we did was we collected African and Asian elephant fibroblasts, and then fibroblasts from species to which they're closely related, right? So in this case, Hyraxes, which is that small thing, aardvarks and armadillos. So aardvarks, armadillos are outgroups to this group. So we got these fibroblasts, they're primary fibroblasts, so they haven't been transformed yet. And then we basically exposed them to lots of things which induce DNA damage. So we dose them with 2 drugs which induce DNA damage or expose them to ultraviolet light. And then we also expose them to other kinds of drugs which induce oxidative stress by increasing intracellular concentrations of reactive oxygen species. And the drug we use for that is TBU, right? So we put all of these cells into a single plate, so it's kind of like a common garden experiment, and then we dose them with drugs and then see how the species respond to those drugs. Okay. So when we do that, we can generate dose response profiles. So that's what you're looking at here, right? So on the x-axis is the dose of the drug. So from 0 to increasing concentrations, 0 to increasing concentrations. And on the y-axis, what I'm showing is the number of cells undergoing apoptosis. So that's shown as a fold change compared to background. And what you can see is that at relatively low doses of different drugs, mitomycin C or doxorubicin, almost no species induce apoptosis. But at sort of small doses, or increased doses of drug, the elephants start to rapidly induce apoptosis. So at this dose, all of the elephant cells are inducing, nearly all of the elephant cells are inducing apoptosis, while the other species are only starting to induce apoptosis. And the same is true for doxorubicin, right? So at this dose of doxorubicin, the elephant cells have started to induce cell death through apoptosis, while the other species haven't. And then what you might notice, for example, is at this dose of mitomycin C, it looks like the elephant cells are reducing the number of cells undergoing apoptosis, but that's actually an artifact of the method, right? So this is a relative method compared to no dose. So all these cells are basically dead, right? So at very high doses, the elephant cells are already dead, or sorry, at relatively low doses. The elephant cells are already dead by the time that the other species of cells have just started to respond. So if they're already dead, more cells can't undergo apoptosis. So these data suggest that elephants have evolved cells to be extremely sensitive to DNA damage. Basically, they don't tolerate any DNA damage. And whether that's through drugs or UV light, right? So elephant cells induce apoptosis at relatively low doses of UV light, while the other cells, cells from other species basically don't respond, right? So one of the ways that we think elephants evolved their large body sizes, or at least a phylogenetic correlation of that, is that their cells evolved to be extremely sensitive to DNA damage. In contrast, if you do the same kinds of experiments, but instead of inducing DNA damage, you induce oxidative stress, it looks like elephant cells have evolved to be extremely resistant to oxidative stress. So at the top graph, I'm showing the LD90, which is the dose at which 90% of the cells are dead in response to that oxidative stressor, TBU. So you can see here that the dose in micromolar is relatively high for the elephants, African and Asian, while it's relatively low for the outgroup species. So these data suggest that African elephants and Asian elephants evolved to be extremely resistant to oxidative stress. So elephant cells are at the same time extremely sensitive to DNA damage, but extremely resistant to oxidative stress. And we'd like to know sort of what are the molecular mechanisms that are responsible for that? How is it that elephant cells evolved to be relatively resistant, relatively more strongly resistant to reactive oxygen species compared to the other species of mammals? Because what we want to try to do is connect, right, make that connection between how morphology evolves and how genomes evolve. In this case, our morphology is large body sizes, our molecular phenotype is sensitivity to DNA damage or sensitivity to reactive oxygen. So we want to be able to connect these dots. So to do that, we take advantage of the fact that there are actually a lot of Afrotherian genomes. So there's a lot of genomes for elephants and the things to which elephants are closely related. So we happen to have genomes from African savanna elephants, African forest elephants, and Asian elephants. So these are the extant elephants. And we also have genomes from some recently extinct elephants. So things like the straight-tusked elephant, which is sort of a very large elephant. Let's see, there's, I think, 6 or 8 woolly mammoth genomes now. There's a Columbian mammoth genome, and there's a mastodon genome. So technically a mastodon isn't an elephant, but it's large-bodied, relatively large-bodied like elephants were. And we also have genomes for things that elephants are closely related to, but that are much smaller, including hyraxes and manatees and dugongs and aardvarks and tenrecs and the like. And actually just recently, like last week or so ago, someone published the stellar sea cow genome. So you can see that stellar sea cow is about the size of an elephant, and we're getting that genome data now to see, if we can identify any genetic changes in stellar sea cow that may be related to large body sizes. Once you have these genomes, then you can start to ask, what are the genetic changes that are unique to the elephants compared to the non-elephants? Because those are going to be phylogenetically associated with large body sizes, sensitivity to DNA damage, and sensitivity to reactive oxygen species. There's going to be a lot of other genetic changes that are unrelated to those things as well. That are sort of related to an elephant being an elephant, or sort of neutral and have no phenotypic effect whatsoever. And we decided to start, at least in these initial experiments, these initial comparative genomic experiments, with sort of the lowest hanging fruit, right? So we decided to see if we could identify gene duplications that were unique to the large-bodied elephants and not found in the smaller-bodied outgroups to elephants. And we identified 354 gene duplications that were found in all of the elephants and mastodons, but none of the outgroup species. So 354 isn't that many, but it's a lot, or isn't that many, but it's more than nothing. So you can start actually doing sort of biology with that. So now that we have these set of gene duplications, we can ask, are these Proboscidean specific gene duplications enriched in particular biological pathways? Do they seem to have similar functions? To do that, we just looked at what pathways and gene ontologies these genes were enriched in. And sort of this is a word cloud showing the enriched pathways. If you haven't seen one of these before, the size of the word is proportional to its negative log 10 FDR. So a word which is about this size has a negative log 10 FDR of 3, right? So G2M transition has a negative log 10 FDR of about 3. And I'm only showing the top 50 pathways. And I don't expect you to see all of these, but some pathways start to stand out, right? So gene duplications in the elephants are enriched in cellular responses to stress. Well, sure, cancer is stressful. Nucleotide excision repair is a pretty interesting pathway to be enriched because DNA damage will eventually cause cancer. So the better you are at repairing your DNA damage, the more resistant you're going to be to cancer. Stabilization of P53 and P53 related pathways are also enriched among those elephant-specific gene duplications, which again makes sense because P53 is sort of the master tumor suppressor. So you would think just from first principles that there are going to be P53 related pathways as you evolve a large body size. One of the major pathways that stands out are all of these. So if you sort of look to see what's in common between each of these, they're all related to cell cycle progression. So if you think about sort of the molecular phenotypes of cancer, cancer is basically nothing except unregulated cell proliferation. So again, it sort of makes sense that in species which evolved to have enhanced cancer suppression, that you might observe augmented cell cycle control. So collectively, it looks like those elephant-specific gene duplications are enriched in the right pathways that might be associated with reducing the probability of developing cancer, and which would maybe allow you to evolve increased body sizes and long lifespans. Which is interesting, but then you want to go away from this list-making activity toward doing functional work, right? Toward doing more reductive molecular biology-like experiments. So now you have to go from your list of genes to actually your list of genes and the associated pathways to say, okay, which of these genes are actually duplicated? So when you do these comparative genomics-like experiments, you get basically an Excel table with gene names, right? So elephants have extra copies of P53, right? So when we initially published this a few years ago, we found that elephants, depending on how you count, have 3 to 19 extra copies. And we have RNA-seq data from a bunch of different elephant tissues, and we could say that these extra copies are expressed and they have coding potential. In this case, the elephant genes are almost all retrogenes. Then there's another duplication of a gene called the LIF. So LIF is really interesting because LIF stands for leukemia inhibitory factor. So if you're interested in the sort of genetic mechanisms that might underlie augmented cancer resistance, a gene called leukemia inhibitory factor really stands out. Then there's a couple genes that are related to DNA, both DNA damage, and DNA repair, like MAD2L1. PCNA is related to DNA damage and repair. Elephants also have a ridiculous number of odorant receptors, so they have several thousand odorant receptors. So those genes come out as duplicated in the common ancestor of elephants. But for the rest of this talk, I'm just going to focus on this gene called SOD1, which stands for superoxide dismutase 1. And it looks like elephants have 2 copies of SOD1, while most other species have 1 or 2, depending on how you count. Right. So SOD1 is a protein that converts normally relatively harmful superoxide free radicals, so superoxide and hydrogen peroxide, superoxides, to molecular oxygen and hydrogen peroxide. So peroxide is not good for you either. That can induce oxidative stress, and peroxide is broken down by catalase. And all of our cells are producing free radicals all the time, just as a byproduct of the electron transport chain in our mitochondria. So if you have mitochondria, you need ways of dealing with free radicals, and SOD1 is one of the genes that do that. So when we look at in a bunch of different mammalian genomes and say how many species have duplications of SOD1, well, it's kind of a lot. So here I'm showing the phylogeny of a bunch of different mammals. And if we look to see the copy number of SOD1, you can see that a lot of species have a copy number that's more than 1. It's just that if you look to see what are the sort of molecular signatures of those genes, they're almost all pseudogenes. So it looks like a lot of different mammalian genomes encode extra copies of SOD1, but all of those extra copies are pseudogenes. And the ones that don't are a few, right? So it looks like bush baby, mouse, sheep, cow, black flying fox, and African elephants potentially encode extra copies of SOD1 that are functional. So they don't have premature stop codons. They don't have sort of signatures of reduced functional constraint. So at least some species of mammals have extra copies of SOD1, including elephants. So we know that at least African elephant has extra copies of SOD1. So what happens when we go back and look in the other elephant and elephant relative genomes to see if they have extra copies of SOD1? So we were able to do this for African savanna elephant, straight tusked elephant, African forest elephant, woolly mammoth, Asian elephant, mastodon, and then a couple of the outgroups. And when we do that, it looks like all of the elephant genomes encode an extra copy of SOD1, while none of the outgroup species do, and that extra copy is a retrogene. So retrogenes are genes which get transcribed into RNA, and then that RNA gets reincorporated into the genome as a byproduct of line 1, right, of line 1 retrotransposition. And we're able to tell that this gene is a retrogene because it lacks introns. So here is the locus for the SOD1 retrogene in the African elephant genome. And all of the exons align up, but none of the introns are present in the duplicate copy. So that allows us to know relatively easily that this is a retrogene. And we also see a lot of the molecular signatures of retrogenes, in particular line 1 mediated retrotransposition. So there's a line 1 target site as a product, a byproduct of line 1 retrotransposition. You get the duplication of these direct repeats on either side of where the retroduplication event happens. And this gene also has its poly A tail that is intact. So right in the downstream part of the 3' UTR, we find a really nice poly A tail. So that's well and good. It looks like elephants and their relatives, like mastodons and mammoths, but not the outgroups, they have an extra copy of SOD1. That extra copy is a retrogene. But the problem is, if you're trying to make these mechanistic connections between having extra copies of things with tumor suppressor functions and then having cells which are resistant to cancer, having a retrogene might not actually help you all that much, because retrogenes usually lack native regulatory elements, which most often makes them a pseudogene, not a functional gene. So what we wanted to know is, okay, elephants and their relatives have an extra copy of SOD1, but is there any evidence that that extra copy is expressed? So what we did was generate really deep RNA-seq from African elephant fibroblasts. So we generated paired end reads for about 150 million paired end reads from African elephant fibroblasts, and then we did de novo assembly. This is way more reads than you would normally need if you're doing an RNA-seq experiment, but we really wanted to know, is there any evidence that this thing is expressed? And when we do that, the black lines that are showing above the genomic locus are the inferred transcripts. So it looks like there is a transcript that we can map back to the SOD1 retrogene, and we're able to do that uniquely because it includes not just the sequences of the SOD1 gene, but also upstream and downstream sequences, which are distinct from the original SOD1 gene. And then when we look in the RNA-seq data, we can actually see, okay, there is evidence that this thing is expressed. So we quantify expression in terms of transcripts per million, which is more or less the molar ratio of a transcript compared to all the other transcripts. And if we look in control cells that we don't do anything to, that's expressed about a TPM of 3. If we treat the cells badly by dosing them with those drugs, which induce genotoxic stress, it doesn't really change expression. If we add that drug, which induces oxidative stress, again, it doesn't really change expression. So it looks like this SOD1 retrogene is constitutively expressed, and we can't seem to induce it with these kinds of drug treatments. So before I mentioned that retrogenes don't normally tend to have regulatory elements. So they don't have promoters and they don't have enhancers because they tend to integrate randomly in the genome. So if this thing is expressed, this retrogene is expressed, then it should have an upstream regulatory element. So what we did was clone the region upstream of the SOD1 retrogene into a luciferase reporter, and then just asked, when we transfect that reporter back into elephant cells, is there any evidence that the region upstream of the retrogene can upregulate luciferase expression? And when we do that, again, we see that there's an increase in luciferase expression, which indicates that the region in red has enhancer or promoter-like functions, but it's not inducible. So just like the RNA-seq data, we can expose those cells to different drugs and not see an induced expression. And the same is true for the promoter region. So we can transfect that luciferase reporter back into elephant cells, treat those cells with different drugs, and sort of nothing happens. There's still basal expression. So again, this starts to sound like the region upstream of the retrogene encodes a promoter, and that promoter is constitutively active. So the next thing we did was design a pair of PCR primers that flank the region that we cloned into the luciferase reporter. And we did a chip qPCR. And for this chip qPCR, we used an antibody for a histone modification that is characteristic of promoters, and that is a histone modification called H3K4 trimethylation. And when you do that, you can see in both control cells and drug-treated cells that there is significantly more H3K4 trimethylation in the region upstream of the retrogene than you would expect compared to the background cells. So the background would be, about 1. So from these data, it looks like the retrogene is expressed. It's expressed constitutively. It has a promoter. That promoter doesn't seem to be responsive to any of the drugs that we treated those cells with. And that region that we think is functionally able to act like a promoter has promoter-associated histone modifications, just like one would expect for an actively expressed gene. Which is all well and good. That suggests that that gene might be active. But what you really want to know is, okay, so you have this duplication of a gene. Does that gene duplicate actually contribute to the increased oxidative stress that we observe in the elephant lineage? So to do that, we did the relatively simple experiment where we took African elephant fibroblasts, and then we used siRNAs to knock down the SOD1 retrogene and treated the cells with that drug, TBU, which induces oxidative stress, and then just assayed cell viability, cytotoxicity, and the induction of apoptosis. So when you do that, here's the percent change in the number of apoptotic cells for African elephant cells that we don't treat with anything except control media. When we add the drug, which induces oxidative stress, there's about a 350% increase in the number of cells undergoing apoptosis, as you would expect. So this is just duplicated here. So we have our increase in cells undergoing apoptotic stress in response to the drug treatment. And if we knock down the retrogene, which is shown here, then we see that there's an increase in the number of cells that are undergoing apoptosis. It's about a 33% increase with a p-value of 0.01 if you sort of care about p-values. So if more cells are undergoing apoptosis, that should mean that more cells are dying, right? So cell viability should decrease as apoptosis increases. So if you look at the control cells that we don't treat with anything, they're usually growing pretty happily, so their cell viability is high. Then if we dose them with the drug, which induces oxidative stress, cell viability goes down. And then if we treat the cells with the siRNA that knocks down the retrogene expression and treat with that drug, which induces oxidative stress, there's a further decrease in cell viability. And that further decrease is about 10%, and then p-value if you care. So it looks like if you prevent the extra copy of the SOD1 from acting, so you reduce the expression of the SOD1 retrogene with siRNA, that you increase the number of elephant cells that are undergoing apoptosis, and that occurs at the same time that there's a decrease in cell viability. So these data suggest that having that retrogene copy is contributing to the augmented resistance of elephant cells to oxidative stress. But that doesn't tell us that that extra copy of SOD1 is sufficient to protect the cells from reactive oxygen-induced apoptosis. So we know it's necessary, but we want to next ask whether it's sufficient. To do that, we just overexpressed the SOD1 retrogene, the African elephant SOD1 retrogene, in Chinese hamster ovary cells. And we picked these cells for no particular reason other than they're not elephant cells and they're really easy to transfect. And then we just did the same experiment where we overexpress the retrogene, treat with TBU, and then assay for apoptosis, cell viability, and necrosis. When we do that, you can see again, no treatment, induction of apoptosis is pretty pronounced in CHO cells when you add TBU. And if you overexpress the SOD1 retrogene, then you protect the cells from reactive oxygen species induced or oxidative stress induced apoptosis. And if you're protecting the cells from apoptosis, then you should see an increase in cell viability. So if you treat the cells with TBU, cell viability goes down. If you treat the cells with TBU and overexpress the retrogene, then cell viability goes up. So this data suggests that not only is the SOD1 retrogene necessary for the full protection of African elephant cells from apoptotic stressors, but we're also able to transfer that to other species cells. So the SOD1 retrogene is both necessary and sufficient for the enhanced protection from oxidative stress that we see in African elephant cells. Oops, wrong direction. So that's cool, but we next wanted to know, how is it that SOD1 is protecting the cells from ROS-induced apoptosis? So to do that, we just took advantage of the fact that we have a bunch of different fluorescence reporters that co-localize to different regions of the cell, right? And so we GFP tagged the SOD1 retrogene, and then we overexpressed that GFP tagged one SOD retrogene in African elephant cells, and co-transfected the cells with a bunch of different fluorescent reporters that either localized to the nucleus, localized to the mitochondria, the ER, and the like, and asked, where do we see co-localization? And that's what these data show. So this is a fluorescence intensity plot. So we take the cells, we overexpress the SOD1 EGFB tagged genes, or the SOD1 retrogene with an EGFB tag, or SOD1 with an EGFB tag. And then we look to see where in the cell those EGFB tagged proteins localize. And the more of the tag that's there, the brighter the intensity. So you can see there's a really strong localization of the SOD1 retrogene with the tag in these sort of distinct spots, which are outside of the nucleus, and not in the periphery. And we see similar co-localization for both the retrogene and the non-retrogene copy. And I won't show the data, but when we look to see what things co-localize there, we find that all of these bright red spots are basically the mitochondria. So it looks like the SOD1 retrogene and SOD1 are accumulating in the mitochondria of the elephant cells, which makes a lot of sense because the mitochondria is one of the central players in the production of ROS, right? So this is where the electron transport chain is happening. So this is where there's the induction of reactive oxygen species. And that happens to be the place where SOD1 is located. So SOD1 is normally located in the intramembrane space of the mitochondria. And it's in the intramembrane space where SOD1 takes reactive oxygen species and converts them into peroxide, and then that peroxide is exported, and then the peroxide is turned to oxygen and water by catalase. So it makes sense that the SOD1 and the SOD1 retrogene are located in the mitochondria. And it also makes sense that it's in the mitochondria because while we know that reactive oxygen species have a whole bunch of normal physiological functions, it turns out that if any of these physiological functions sort of is taken to the extreme, that they induce apoptosis. So if any of these normal physiological functions for reactive oxygen species goes awry, then we induce apoptosis. So it sort of makes sense that you see the mitochondria and SOD1 being related to apoptosis. So we next wanted to ask, okay, does having that extra copy of SOD1 protect the cells from mitochondrial reactive oxygen species? And to do that, we did a relatively simple thing, right? So we took the elephant cells and we knocked down the SOD1 retrogene, and then we treated with that drug TBU. And this time we assayed for mitochondrial health, and we did that using a drug called mitosox. So mitosox is an indicator for superoxide. And the way this drug works is you sort of have cells growing in culture, you give them mitosox, which is what these gray dots indicate. They cross over the plasma membrane, and then once they get into the cytoplasm, they accumulate in the mitochondria. And as a byproduct of that accumulation, they start to fluoresce green. So happy, healthy mitochondria fluoresce green. Mitochondrial dysfunction leads to holes forming in the mitochondrial wall under these pores opening. And when that happens, you sort of lose the mitosox indicator from being accumulated in the mitochondria. So you lose green fluorescence. So in this case, green mitochondria happy, not green mitochondria dying. So when you do that, you see results that look like this, right? So if we knock down the SOD1 retrogene and then assay for mitochondrial ROS levels, in this case, that is proportional to how healthy the mitochondria are, you can see that if we have a control media, the cells are pretty happy. If we induce reactive oxygen species with that TBU drug, then we get about 106% increase in the number of apoptotic cells, or the number of unhealthy mitochondria with significant p-values, if you care about that. If we knock down the retrogene, then we see a significant increase in the amount of unhealthy mitochondria, which indicates that the SOD1 retrogene contributes to the maintenance of mitochondrial functions in the face of free radical production. And if you wait a really long time, so you go from 8 hours to 24 hours, if you knock down the retrogene and treat with that drug, you almost completely lose fluorescence, and that's because the cells are almost entirely dead. So 24 hours after treatment with TBU, the cells are starting to recover. So very, very unhealthy cells still starting to become more healthy and recover from TBU treatment. If you knock down the extra copy of that SOD1 retrogene, the cells are unable to recover and they basically all die. So it looks like the SOD1 retrogene protects cells from oxidative stress-induced apoptosis by reducing mitochondrial reactive oxygen species, which makes sense given what SOD1 does. So this is not too surprising because we know that the mitochondria is a focal point for many of the programmed death pathways. So lots of pathways, all of these pathways lead to programmed death, not just intrinsic apoptosis, but all of these other ways that cells can kill themselves, including necroptosis and pyroptosis. And one of the central players is the mitochondria. Which leads to a question that we can't really answer, right? So we really want to know is, does having an extra copy of SOD1 really protect against cancer and increase lifespan? And we're not going to be able to make transgenic elephants, although that would be cool. So we're not going to be able to knock out SOD1 from elephants or knock in a duplicate copy and ask, is that protecting cells, protecting elephants from cancer, or if you knock it out, predisposing them to cancer? Because elephants live a long time and you're never going to get approval to do experiment. So we can do the next best thing and we can ask, does having an extra copy of SOD1 protect other animals from reduced cancer incidence or increasing their lifespan? And for some things, it seems yes. So if you overexpress SOD1 in melanogaster, you can extend their lifespan. And if you do selection experiments and try to select for extended longevity in melanogaster, one of the byproducts of extended longevity is increased SOD1 production. The same is true in yeast. So if you overexpress SOD1 in Cerevisiae, then you can increase their lifespan. And if you treat C. elegans with drugs that mimic the function of SOD1, then you can increase their lifespan by about 50%, which is kind of neat because that suggests that you may be able to turn these drugs into sort of human lifespan extending drugs. But it's biology, so it's complicated because of course it is. So in an ant, which has really large differences in body size, this ant is Lasius niger, the African black ant. The queens in the center, they live for almost 30 years, and the workers, which are the ones around her, they only live for about a year or two. And the sort of males, which these are all haplodiploid, so the males live only a few weeks. And if sort of what we've taken, what we would expect is that the females, which live a long time, should have a whole lot of SOD1 expression. And it turns out that that's not the case. Queens have about 75% less SOD1 than the workers and the males. So they're an exception. And you can try to do the same experiments in mice. So if you overexpress SOD1 in transgenic mice, you find that those mice have a shortened lifespan and they mimic some of the effects of Down syndrome. And that's because Down syndrome SOD1 is found on chromosome 21. So people with Down syndrome have an extra copy of SOD1. And there are all these deleterious consequences of trisomy 21, including at least some of them, which we think are because of the extra copy of SOD1. So if we ask, does having the extra copy of SOD1 actually reduce the risk of cancer and increase lifespan? Well, it's complicated. Maybe it does, or maybe it doesn't. It's probably because having extra copies of SOD1 imposes some cost. And unless you can find a way to pay that cost, then you're not going to be able to have extra copies. So elephants have extra copies of SOD1, so they must have found some way to compensate for the deleterious consequences of extra copies of SOD1. So in conclusion, it seems like elephants are nearly invincible. And they did that at least potentially, or at least partially, by evolving cells that are nearly intolerant of DNA damage, yet extremely resistant to reductive oxygen-induced stress. It looks like genes with tumor suppressor functions and anti-ROS functions increased copy number in the common ancestor of elephants, at the same time that large body sizes and long lifespans were evolving. At least one of these duplicates, which is the SOD1 retrogene, promotes resistance to mitochondrial generated reactive oxygen species and oxidative stress. And it's probably the case that lots and lots of genetic changes contribute to the evolution of longevity and cancer resistance in elephants. And the SOD1 duplication is just one of those. So we have to go and find them all if we want a causally complete explanation for why elephants were able to reduce their risk of developing cancer. And with that, I'll thank people who give us money, people who do the work, because while I go into the lab, sometimes I barely do it anymore. And I'll thank you for listening. And if you haven't seen an Hyrax before, these are cute little Hyraxes. And with that, I'll take any questions if you have any.
Thank you very much.
Only a minute
That was
over.
great. No, that was great. Hold on. We're trying to turn off the thing so I don't get double sound. Okay. Yep. Look at those mini elephants. There they are. All right. So
Adorable.
that was fantastic. That was great. And I really enjoyed it, and also thought you did a very good job of actually explaining things. So we'll see if we have any questions. No, it was great. It was great. Let's see. So we have a couple questions about kind of mammals. So it's two-part. Like maybe someone heard you say that there are maybe 2,200 species of mammals, but they thought there was more like 5,900. Okay.
Yeah, there's 5,900. If I said 2,200, I misspoke.
You misspoke.
I think I meant we almost had 2,200 in the tree.
Oh, that's I think, yeah, thank you. Any information on cancer rates in the chrysochlorids?
Oh, no, that would be cool. So no, and the reason for no is that they're really hard to keep in zoos.
Those are the golden moles and things?
Yep, exactly. Yeah. Those are the golden moles. Yes. So we don't have a lot of golden moles in zoos, so we're not able to sort of determine whether they have cancer or not. If an elephant dies in a zoo, they get really sort of complete pathology done on them. So that's how we know what their cancer incidences are.
Yeah. Is the SOD1 RTG artificially put in the hamsters, or do they have any of that naturally occurring, or is it just a complete add-on for them?
So hamsters have one functional copy of the SOD1 gene.
Okay.
They have a non-functional pseudogene, which has all sorts of frame shifting mutations. So it's probably not functional. And then we overexpress the elephant in that one.
Yeah, okay. Another question, would that extra copy of SOD1 potentially increase ALS risk in these elephants? I think ALS is linked to SOD1 mutations in humans. Was wondering if something similar could occur in elephants.
Yeah, so obviously it's biology, so it's complicated. So it's not just having an extra copy of SOD1 that contributes to ALS in humans, it's having the mutation that contributes to ALS in humans. So if you have a mutation in SOD1, which prevents it from sort of doing its thing, that causes death of neurons. So elephants just having an extra copy probably doesn't predispose them to ALS, but it might protect them from ALS.
Yeah, if they have a couple copies, they have to get 2 mutations.
So then just like cancer, you got to get multiple hits.
Right. Very interesting. Okay. 2 more at least in the chat. Can you let's see, can you maybe explain the link between SOD1 and cancer protecting from apoptosis could also favor cancer and uncontrolled proliferation, even if reducing ROS effect can of course protect cells from damage? Do you think SOD1 can explain cancer resistant, or is it maybe a different mechanism?
So I think it's a different mechanism, most likely. So if you induce DNA damage in the elephant cells, they basically don't tolerate any of it. So their sensitivity to DNA damage is low. One of the things that we know that ROS does is induce DNA damage, right? So if you increase ROS, you increase DNA damage. That should increase apoptosis. So if you protect cells from ROS, then you're probably protecting cells from that increased DNA damage. But we know that DNA damage and sort of the damage that ROS does accumulates over time as a product of aging, right? So it's complicated. It probably protects cells from DNA damage and also helps those cells be better agers.
Yes, and then one more question right now. Could the retro copy, got another one. Could the retro copy have a different activity that could explain the results seen in mice, which overexpress SOD1? Do you plan to, go ahead,
I was going to say at least
and then
with
bats,
regard...
and then I said, what's going on in bats?
Oh, of course, bats.
Yeah. My lab is OneNote, man. But yes, go ahead.
So the SOD1 retrogene is identical at the amino acid level to the SOD1 gene. So if there is a difference, it's got to be in an mRNA stability or rate of transcription or something. We don't know what's going on in bats with regard to the retrogene. They have some pseudogene copies. Whether those pseudogene copies are actually expressed and functional or not is not clear yet.
Cool. Is decreased ROS production in flightless birds related to their larger size or their ability to be larger?
So we don't know. The weird thing about birds is they don't seem to escape Pettos paradox. Right. So if you're a mammal and you're big and you live a long time, that's associated with being cancer resistant, but birds don't seem to have any correlation between body size and, birds seem to have a correlation between body size and cancer rate, such that the big birds get more cancer than the little birds. So whatever's going on in birds is completely different than what's going on in mammals.
Huh. Do elephants and other large species die from diseases other than cancer more than you would expect? Seems like this cancer protection is a too good to be true benefit without any trade-offs.
Oh no, there's definitely trade-offs.
Okay.
Like there's trade-offs for everything, right? So
Yeah.
elephants do die from other things, but the way they die from other things is different, right? So elephants get heart disease, but they get heart disease slightly differently than the way humans get heart disease, right? So they seem to suffer age-related decline in fitness, just like we suffer age-related decline in fitness. Elephants die a kind of horrible death, actually. So elephants have 3 sets of teeth. And once they go through the final and third set, they can't have nothing to replace them with. So they're not able to eat. So if they don't die from being preyed upon and they don't die from some other mechanism, then they starve to death. So they can live a really, really long time. It's just that they're still dying from things. Extrinsic things. They still have extrinsic mortality.
Yeah, I knew they did that with the replacement, but I didn't know what the fundamental cause of death was. Last question from Clive, who's talking to you later. Do elephants have more catalase to handle the extra peroxidase made from extra SOD1?
Ask me that in a couple of weeks when we're done measuring it. So it's
a good question. Good question.
That is a good question. The other thing is that catalase is the fastest and most efficient enzyme that we know of in mammalian genomes. So we probably haven't maxed out the ability of catalase to do its thing.
Cool.
Maybe
well, thank you so much. Thank you so much again for coming to speak to us. And again, for those of you who saw this, or if you have other people that might want to see it, it's going to be posted. So you can watch it through the end of the quarter. So thank you very much.
No, always happy to come and talk.
Bye, everybody.
Have a good day, all.
Thanks. Bye.